Hey, everybody, it's Dr. Eric Balcavage. We're back for another edition of the Thyroid Answers podcast. We have another guest today. His name is Ricardo Miranda. He is a functional medicine practitioner and a researcher on MTHFR gene mutations. So we'll be talking to him about what the heck MTHFR is. It's not a curse word, by the way, and what it has to do with health, well-being, thyroid physiology, and some crazy thing called methylation. So Ricardo, welcome to the Thyroid Answers podcast. How are you doing today?
Oh, very good. Hi, Eric. Thanks for having me. Great pleasure to be talking to you here today.
Sure. So maybe give the listeners a little bit of background on you. Who are you? Why should we listen to Ricardo Miranda about MTHFR?
Well, I'm the CEO of MTHFRdoctors.com, which is a website that educates people about MTHFR. But my background, I was trained in Chinese medicine and been practicing for 30 years. I incorporated functional medicine in my practice, and I dove into the field of epigenetics. And well, if I may, I'll give you a little story how I got involved with that.
Sure.
So I went to years ago to a lecture on epigenetics, and I'm always interested in longevity. And I found out that our DNA is not our destiny. And years ago, 40 years ago, people used to think that if you had in your genes from your family some specific diseases, that inevitably you would develop that because it's in your genes. But I found out that no, not necessarily. We can actually control the expression of our genes. And I was fascinated by that and start researching and I want to find out how I can control the expression of my genes. I'm interested in longevity. And I found out that there's a process called methylation that turns genes on and off. And that's the process that controls the expression of the genes. So I was very interested and continue researching. And there are 26 genes that affect methylation. And the most important one of them is the MTHFR gene. So that's how I got into the MTHFR and continue developing my studies and research on that, and got into some experts in the field that Dr. Lynch, and I realized that you and I met briefly in 2015 at a conference I went to on epigenetics and methylation, as Dr. Lynch's conference that I was attending. You were a speaker on that conference.
I was.
So it was very nice experience. So that's my involvement, and I continue developing the website with the intention of educating the public about MTHFR, how important MTHFR is, and so that's my small intro on my involvement.
Perfect. So let's talk about what is MTHFR, because some people don't know what that is. It sounds like a bad word when you say it. So what is MTHFR?
So MTHFR stands for methylene tetrahydrofolate reductase. It's a gene that controls how the body breaks down folate and folic acid, for example, which there's a difference. They're not the same. So folate is the main fuel for methylation. It's the number one fuel for methylation. It controls methylation. That's why I consider MTHFR the number one gene for methylation. And over 80% of the population has a mutation on the MTHFR. So depending on the mutation that you have on this gene, you may lose from 20% to 70% of your ability to break down that folate or folic acid. And when that happens, and then it will affect your ability to detoxify. Methylation controls intercellular detoxification. So it has a direct effect on how people detoxify, for example. And it's related to many different health conditions as well. So it's a very important gene, and because the prevalence is so high, everyone should really know if they do have a mutation or not on this gene.
Okay. So it's a gene, you say maybe 80% of the population has some genetic variation. And for the listeners, when we talk about genes, there's different forms of the gene. One of the genes that when the gene is in its most common form, we call it the wild type. And then if there's alterations in the amino acid sequence of the gene, we can call that a polymorphism. And that can alter the function. And that's what he's talking about with that MTHFR polymorphism. So is there just one type of MTHFR polymorphism or alteration, or is there more than
one? There are many types we call alleles on the MTHFR gene. However, we mostly look at the two main ones, the MTHFR C677T and the MTHFR A1298C. Those are the most important. A couple other ones that, depending on what condition we're looking at, they are, they are important as well, but mainly we look at these two.
Okay, and this gene plays a critical role in, as you said, in the conversion of dietary folate into its different metabolites. And tell us a little bit about that. We call that the folate pathway or the folate cycle. Exactly. What is folate? So what does it do? And why does it need this MTHFR gene? Why is it so important? What is that MTHFR gene doing to that folate to make it important?
So folate is found on foods, mostly leafy greens, broccoli, spinach are rich in folate. And folate goes through several processes of breakdown. And the MTHFR gene controls that process. So it breaks down from tetrahydrofolate to 5,10 methylene tetrahydrofolate. It goes to 5L tetrahydrofolate. I'm being very brief on that. It's a much more detailed breakdown. But there are several levels of breakdown that is controlled by this gene. And when you have a mutation, you lose your ability to break down efficiently. So for example, if you have the homozygous, which is a double mutation on the worst allele, we call it a homozygous mutation on the MTHFR C677T, for example. So that person will lose roughly 70% on their ability to break down the folate into the usable form of folate, okay, which is the 5-val methylene tetrahydrofolate. Okay, so what happens? The folate helps to transform homocysteine into methionine, for example, and you have and with the help of B12 as well. And that is it makes the wheel of methylation turn. So one of the most common let's say side effects of this breakdown or lack of the breakdown of the folate or the mutation of the MTHFR folate is that since you don't have enough of this fuel called folate to transform homocysteine into methionine, the buildup of homocysteine in the blood rises, and the patient has an elevation on homocysteine in their blood. So homocysteine is a amino acid that could be inflammatory, specific for the heart. So cardiologists, they know well about the MTHFR because of homocysteine. So that's one of the consequences of this mutation, for example. And it can affect your cardiovascular function and so forth. But methylation, if you don't have enough, if you don't transform your homocysteine into methionine, you don't have the wheel going on, you're not going to methylate well. The methylation is going to be sluggish. So the person will be be undermethylating. And that will affect, for example, as I mentioned, their ability to detoxify, for example, and it affects many other conditions. We'll get to the thyroid later. But this is one of the most common consequences. MTHFR directly affect mental health, for example. So a lot of patients with MTHFR mutation, they end up having more anxiety, for example. There's a correlation between MTHFR and anxiety. It's not the only gene for that, but there is a correlation. So with that said, anybody that has a mutation on MTHFR, they should avoid, never take folic acid. Because as I mentioned before, folate and folic acid are different. And folate, if they can eat folate, they should eat folate. They should, you can have folate from food, that's okay. They're not going to absorb all the folate from food. So if they have that mutation that they lose 70%, when they eat their broccoli, they might be absorbing 30% of the folate that someone that doesn't have a mutation would. But that's okay. But if they do take a supplement, for example, containing folic acid, what happens is not only they won't absorb the whole thing, but because it's synthetic, the unmetabolized folic acid becomes toxicity. So it gets stored in the space, it's called the intracellular matrix, that stores toxicity in our body, and then our body needs to detoxify. So it's very important.
So for the listener, folate, when we're talking about folate is B9, okay? Just so everybody knows what we're talking about. And it's in plants, it's in food, it's in our food source. Once you eat food, break it down, and it gets absorbed into the body, then that folate needs to be converted into other forms, folinic acid, methylfolate, different forms of folate that are usable by different cells, tissues for certain actions in the body. So it's really critical that not only we get sufficient folate, but that we actually convert it into some of the usable forms. And this gene, which then produces an enzyme called MTHFR, then does this step in the reaction that converts folate, this dietary folate, in one of the forms that's really usable to support this thing called methylation. And we're going to talk about what methylation is here in a minute, but I don't want to get too far, but I want to make sure we cover those things. But I want to make sure, I heard you say something and I'm not aware of this, so I just want to make sure, having MTHFR gene mutation, are you saying that impacts the absorption of folate from our diet? Or is that enzyme, the gene and the enzyme it produces, impacting the folate once it's already absorbed into the body and it's impacting it at the cell level?
Yes, impacts the breakdown of the folate into the usable form of the folate.
Right. So it's already absorbed into the system and then it gets converted, right? Because I didn't want somebody to think, oh, I can't absorb it because I have this MTHFR polymorphism. You can absorb it.
Correct. Thanks for clarifying. That's good.
Sure.
Yeah.
So when we think about folate and we say, hey, this could be affecting maybe 70% of the population, and you said, hey, it could create a lot of symptoms. It can have an impact on your detoxification pathways. It can have an impact on your ability to do a whole bunch of chemical reactions. It has an impact on neurotransmitter physiology. So it has wide ramifications in overall physiology, and it could be really important. But how would somebody, aside from having lots of symptoms, which somebody might say, well, that's just anxiety, that's just depression, that's just inflammation, that's just whatever, how would somebody go about determining if they have folate deficiency or a genetic polymorphism like the MTHFR polymorphism that's impacting their folate physiology?
Yes, that's a good question. And because many of these symptoms have multiple causes, like most health conditions they have multiple causes.
Correct.
So the only way to really know if you have that is by testing. You need to test to see if if you have a mutation or not, and you need to do blood work and measure your folate levels to find out if you're folate deficient, and then you calculate your folate levels based on your mutation. Because when you test for serum folate, and you're testing for all kinds of folate, including unmetabolized folic acid, and there will be some methylated folate as well. But we have in all types of breakdown you have on the blood work. So when you test for serum folate, you don't really know if your body's absorbing everything that you measure. So if your folate, which most labs consider above 5.4 as being normal on folate, my experience, I would say the functional level of folate should be 10 or above. But let's say you do blood work and your serum folate is 10. You're like, okay, so I have enough folate. Well, maybe not. If you don't have any mutation, you'll be able to break down that folate and use it. But if you have the homozygous mutation, for example, and you're losing homozygous mutation on the MTHFR C677T, because you lose 70%, because you can have a homozygous mutation on the other allele, and you lose about 40% of your ability to break down your folate. But let's go back. If you have the worst mutation, you measure your folate, it's 10, but you losing 70% of your ability to break down the folate. So at the worst case scenario, you would be at a 3. You're not really at 3 because there will be some methylated folate on the serum folate as well, but you definitely don't have a 10. You're not absorbing 10. You're not using the 10. So you'll be below your the useful form. So whatever is the number that you consider normal, even if you want to go instead of the functional level, if you want to go the lab normal, and if you get that, and if the lab says 5.4, and if your test came back and you have 5.4, or if you have 6, well, you may not have enough anyway. So I like it to be at 10 or little or above, actually a little more, to have a cushion, because the folate is constantly changing as well. Did I explain well?
Yeah, I think so. So for the listener, when we're measuring folate, and I think we may differ a little bit on this, when you're asking your doctor to measure your folate level, they're typically running a serum folic acid test. And essentially what they're measuring, it's based on the work that I've done and talking to people that actually do the testing. We're typically measuring more of the folic acid, the oxidized folate, when we're doing a serum test. And if that's the oxidized form of B9, that's not a very usable form. That's a form that maybe already been compromised. The traditional lab range would tell you it needs to be greater than, they'll say greater than 6 or whatever, but that's not really measuring the usable form, which I think is what you're trying to express.
Yes.
And my friend Ben Lynch, I think he would tell you we don't want that level to be over a certain level. If we're actually measuring folic acid, we don't want it over 10. We actually would want it less than 10. So what are we measuring? And so from my experience, what I've been told is that if we're really going to take a look at folate levels, we really need to be looking at something like RBC folate. What's actually
folate.
The amount of folate actually inside the cell, because what's outside in the bloodstream may already be oxidized. And so if you're going to your doctor to say, hey, I think I want to know if I have a potential folate issue, or they're testing folic acid, that's probably not the best way to determine what your folate status, your B9 status is inside the cells. And the other thing it's not doing, it's not giving you an idea of how well you're converting that folate into the different types of folate, folinic acid, the methylated folate. You need to get either an RBC folate done, which you can get at your traditional lab, or you can go to some specialized labs. One of the ones that I really like is HDRI Labs, which does a lot of methylation testing, which is something we're going to talk about, but they test this whole thing called the folate pathway or the folate cycle, where they measure all the different steps and all the metabolites inside the cells of folate, so we can see how well how much folate's in the cells, how well do you convert it, and where might a breakdown problem be. So I see so many people, and I'm sure you do as well, that say, hey, my B9's fine, look where it's at. I'm like, that's essentially you're measuring folic acid. We don't really want that high, but you may still have a folate issue. And the best way, in my opinion, sometimes to look at that is to look at one of those more specialized breakdown tests, or we can look at sometimes very simple markers of folate deficiency. We can look at red blood cell indices, can give us an insight. We can look at, well, like if white blood count is low, that can be a folate issue. So there's a number of labs on a general lab panel that if we do a good job interpreting, we could say, yeah, you've got indicators in your general labs of potential folate deficiency, and then we can say the folic acid may be high, but the tissue level, the other tests indicate that there's not appropriate tissue status. Would you agree with that?
Absolutely, absolutely.
All right.
Is that most people they don't have the ability to, or they are not going to doctor. They're doing the RBC folate. They just have the regular folate serum folate levels. So if they calculate based on their mutation, they'll have a pretty good idea.
So yeah, and you're going to see, for the listener, if you're getting labs done, you're trying to get your B9, B12 tested done, ultimately what your general practitioner is going to do is that serum folic acid, they're probably going to do serum B12. And both of those are not great markers of what's happening inside the cell. The most important thing isn't necessarily what's in the bloodstream. It's what's inside the cell and how that's working. And that's where we're going to get into this whole methylation cycle and the detoxification, because all the magic is really happening inside the cells. And it's not necessarily just about what we consume.
And
so
that's why somebody needs, they need to seek somebody like Dr. Balkovich here, because then they would have the right, the right knowledge to go at the right test to get the right information. Yes.
Absolutely. And that's one of the reasons probably why you created the website. So if people need more information on folate physiology, they can go to the website. And I saw you have a list of some doctors on there. Many of them I know through different conferences and seminars and meetings.
Yes.
So let's talk about, we talked about this MTHFR gene. If somebody thinks they might have a folate issue, they want to know if they have an MTHFR polymorphism. How do they get that done? Obviously it's a genetic test, but can they go to their primary care? Can they go to a regular lab? Do they need to go to a specialized lab? How do they get that done? And then I'll give you part two of that question, but how do they get the testing done?
Sure. Well, all of the above. They can, ask their doctor, go to, to send them to any lab. They can order through, for example, our website. We have the cheek swab. It's very, very convenient. They don't need doctor prescription for that. They can order that, do the cheek swab, send to us. They can ask their doctor and go to a lab. The most important thing is to get it done, get the information.
Okay. And so if they can get the test done, and for the listener, there's a lot of different companies that do these cheek swab tests. 23andMe is one of the companies it was one of the early companies that came out and did these things. And I don't know that they were doing this necessarily from a health perspective in the early part, but maybe they were, but they were looking at
stuff. Yeah.
Yeah. But now there's lots of different companies, and all of them are a little bit, some companies have their own chips and they measure a little bit different things on them. And typically if you order the test through your general practitioner, they can do it for you. Maybe it's covered by insurance, maybe it's not, I don't know. But having the result is one thing. Knowing how to interpret what that result means with your signs, your symptoms, your other labs, and maybe the rest of a genetic panel, if you had more than that done. If you go to your family practitioner, they may run MTHFR, but they may not look at the rest of the genes. And you've got lots of genes that code for lots of enzymes that affect the folate cycle, the methylation cycle, and all these. And we got hundreds of them, not hundreds, thousands of them. And so it's important that if you get the testing done, but then the key is what does it mean? And that's a really important thing. So once somebody has, um, do you typically recommend that they just do the MTHFR gene, or do you typically recommend that they do a more advanced panel so you can take a bigger look at the broader scope of what might be going on?
Okay, that's a very good question. There isn't only one way to achieve the results. So in medicine, what I have discovered is that there are several ways that we can take it, that we can achieve the results that we want. Before we developed this test, I actually started out by developing a software to decode 23andMe raw data. So we still have that. So we have different, if people have done 23andMe, for example, or Ancestry, they can go to our website and order these tests, and we have a whole panel for methylation. We get all the genes related to methylation. We have another panel that gives like 600 different genes. We look at, so we have different panels that I developed. I have a background in computer science, so I wanted to buy a software that would do that, but I couldn't find, and then I ended up developing my own. So I wrote the program to develop that. So I have different panels that look at different health conditions, for example, that you have mutation towards it, and it's all broken down and very easy to see. So you can, yeah, and you have the methylation panel as well, complete panel. So you can look at that, look at all the genes, but also look at the mutation you have to possible conditions, and then work with your healthcare practitioner to optimize your health. And most health conditions are controlled by multiple genes. And so when you look at that, it's not like one gene that will cause that condition or that disease, except for a few, like sickle cell anemia. We got the BRCA genes, the breast cancer. There's a, you got that one gene, your chance of having cancer is, I'm not sure, 88, 90%. So, we can't do much about it. But most health conditions, mental health, cardiovascular disease, diabetes, most cancers, they're controlled by multiple genes. And that's where methylation comes into play. So once you identify what predisposition you have for certain health conditions through the panel, then you can optimize your methylation, and I always say optimize your gut. I know you're also an expert on gut as well. So you optimize gut, optimize methylation, and then you can control gene expression. So at first, years ago, I started looking at that. Now, my clinical experience, is that I realized that to control methylation efficiently, I prefer to address first MTHFR and maybe COMT. I look at 2 genes. I don't look at all the genes affecting methylation, because every nutrient that if the gene has a mutation, it affects the biochemical cycle. In the biochemical cycle, each biochemical cycle needs specific nutrients to make it run. That affects that. So when you look, my preference in my clinical experience, what I have found is that when you give a specific nutrient to affect a specific biochemical cycle, and that's something that I remember from that conference that you spoke. There was a doctor that he mentioned that. I don't know if you remember that. He showed a picture, an image on the big screen with hundreds, maybe thousands of biochemical cycles. And then on the corner, he drew a little red circle on a small little piece of corner. And he said, you know what that is? That's the methylation cycle. It's a tiny cycle compared to all the biochemical cycles affecting our health. And each nutrient not only affects the biochemical cycle that we intend to, but it also affects 200 others we are unaware of. So what I have found is, if we just look at MTHFR and COMT for methylation, and you optimize the gut, for 90% of the time, everything comes into play. Now, and 10% that we need to look into, and then we go and find out what else we need to address. And for me, that works very well. Now, different practitioners, they have different preferences. That's okay too. There's some people that are very comfortable looking at different genes and then working with the different cycles and prescribing the right regimen, and it works perfectly for them. So that's fine. So there are different ways that you can achieve results. I prefer to go small and look at MTHFR and maybe COMT, and then look at the gut, then optimize. And sometimes I do cortisol as well, because cortisol will affect methylation. Research has shown that, and vice versa. And methylation can affect cortisol as well. But as far as the genes, I like to go MTHFR and COMT only, and gut. I always combine it with gut, because gut is so important.
So I kind of want to kind of simplify some of what we just covered there. I think for the listener, one of the things that's really important that you said, and the person you were referring to was Neil Nathan. And
yeah, that's right. Yes.
The challenge in functional medicine sometimes is we learn something and then we get on it, and then everything is about one thing and everybody gets overly focused on it. And the challenge is when we do genetic testing, and genetic testing for some of these things kind of goes through as ebbs and flows. I got really into this with Ben Lynch, and that's what led to all those conferences that we were doing. And but the challenge is that some of these, if you decide you want to get a genetic panel, and these have become very popular and people getting these done, and I think the concerns that you want to have when you're getting these genetic panels done is how these things are interpreted. They're different. People have different philosophies about how to address it. It doesn't mean what I say is right or what you say is right, but everybody's got different philosophies. But what you're going to get from some of these reporting systems is you're going to get a lot of genes typed out, and then you're going to get this long report. And what Ricardo is saying that, hey, there's vitamins, minerals that support the function of. So the gene is the genetic material that encodes for an enzyme, and the enzyme then needs to do an action, and that enzyme requires cofactors to work, and that's where the vitamins and the minerals come in. And when you get a genetic report or panel, somebody may say, well, you have this gene, the polymorphism, so it's not going to work as well. And there's two things that are going to influence how well a healthy gene works or a wild type gene works. The more common type of the gene works, and there's things that can make it work better, and some things that can inhibit it. And when you get some of these genetic reports, what happens is somebody will tell you, hey, these are the cofactors that help. You have a polymorphism. These are the cofactors, the enzymes, the vitamins that help it work better. So you should take this because you have a genetic defect. And so then people wind up buying lots of vitamins and supplements and minerals to treat the 10, 20, 30, 40 different polymorphisms that they have. And as they read through the report, especially on their own, they may find out that, oh, this gene says I need to take B6 because I have a polymorphism, but this polymorphism down here tells me that B6 slows down that gene, so I shouldn't take B6. And now there's this host of confusion. And I think that's one of my biggest problems with the reporting systems that are out there and people ordering these tests on their own.
Absolutely.
Two, there's a lot of genes that are on a lot of these reports that are nonsense genes. They don't do anything. They're not functional.
They're no relevance. Yes.
But right. So we wind up focusing on something that maybe means nothing, right? As you said, there's lots of MTHFR polymorphisms. Some have meaning, some do not. But knowing that you have these things is important. But I think it's important that if you are going to dive into looking at your genetics, there's a couple things that we need to consider. This is something that you really should work with somebody who's a skilled practitioner, not just somebody who just runs a report and then sends you a report for, hey, I'll send you a kit, I'll send you a report for $150, $200, and you can, it'll have everything on there. That's not that's not the way to do this, and that's going to lead to confusion and a lot of money spent and not be well. If you work with somebody who's a clinician, they can look at these gene polymorphisms. They can look at a report and say, look, it does look like you have a polymorphism, but it doesn't seem here like it's creating a problem. Or you don't have a polymorphism of this gene, but that's where there is a physiologic breakdown, and we need to impact that. And we've had Ben Lynch, he's on the podcast before, and Ben has written a book called Dirty Genes. Yes. And what Ben talks about in that book, he only gets to cover about 7 of the genes because of the way books go. You pick the 7 sexy ones and you do them. But what Ben's philosophy, it's my philosophy, I'll get the idea of whether it's your philosophy. But when we're looking at some of these genes that may have polymorphisms, sometimes it's my philosophy, before we rush to provide the cofactors, the things that should make it work better, we want to make sure that we're addressing the things that may be inhibiting that enzyme. Because if we're just adding more stuff, but there's something inhibiting it, we may not get the desired effect we want. And the other part of that is what you brought up with Neil Nathan. Just because you have an MTHFR deficiency, a polymorphism, and somebody says you need to take X or Y or Z supplement because of that, we're going to give you methylfolate because you don't make it well. Just because we give you methylfolate to maybe support that pathway, or maybe to support your methylation cycle, it doesn't mean that that's what the body is going to use that vitamin or nutrient for. Just because you have a gene polymorphism that B6 makes that enzyme work better, doesn't mean because you take B6, it's going to go to that pathway. The cells and the tissues get to determine what's most important to them. Absolutely. And what I learned from Dr. Robert Navio's work, who you may be familiar with on the cell danger response, he listed out how micronutrients are used differently. If I'm in a low stress state, I may use a micronutrient to make me feel good. But if the cell's in a stress state, it might use that micronutrient to do more defensive work, which may drive more inflammation because that's the appropriate strategy at the time. So I think the idea when we get these things is we want somebody who's skilled in looking at these in line with your signs and symptoms, in line with your other blood work, to say, yes, there is a problem with your methylation cycle. You do have a genetic polymorphism. Maybe before we start jamming you with some methylfolate, maybe we better make sure that we address some of the things that may be causing that gene, as my friend Ben would say, to be dirty in the first place. Do you agree with that thought process?
I agree with everything you said. And that's why on our reports, if they do the raw data, we do not recommend any supplement. We give the reports. And then what we do recommend is that they consult with an expert in the field to guide them, because as you said, it gets complex to practitioners. It gets complex to doctors. It's a very complex issue. So they should not be searching on their own on what to do. It's a very complex issue that they need guidance. So they need somebody with expertise to guide them on that. And that's why on the reports we don't. And then we developed the test for the MTHFR and COMT, which I prefer. But most of the time, when I see patients, I'm telling them to stop taking stuff. The majority of the time, I'm not prescribing supplements. I say, cut it, because they're just overtaking. The people, they read on the internet, I should take these, and exactly what you said. And then they're taking a bunch of things that the body's not going to utilize the way that we intend. And I had one patient that he was taking 95 pills. He had 95 bottles a day, 95. So I took a picture of the whole table. So the whole thing was reducing, and he did the test with a different practitioner for heavy metal toxicity. and micronutrients. And then they brought it to me. So all I did was reduce from 95 to a half a dozen. We worked the gut, and we did a couple of things with the patient, but as far as supplements, no, when I did acupuncture, I did some. 3 months later, I sent them back to the other practitioner. They did the test with them again, and the heavy metals, everything came down to normal. The micronutrients, everything was deficient. Everything rise up, everything stabilized. So we're able to stabilize without giving more. So we just remove things and basically optimize the body. And as you said, the body will know where to put the nutrients.
Right. And I think that's key, right? So if you have one deficiency, right? If you have let's say somebody does some labs and they say, hey, it looks like you might have some folate deficiency. Could you have MTHFR? Sure. Should you be taking folate? What type of folate, right? That's one thing. But if you have multiple micronutrient deficiencies, you don't have a supplementation deficiency. What you have is maybe a poor diet, poor digestion, poor breakdown of foods, poor absorption, poor assimilation. And then we have to start asking why that occurs. So I agree with you. When you see, one of the first things I tell clients often, if they need to take 5, 10, 20, 30, 40 supplements per day and still feel lousy enough that they need to call me, then they don't need to take any of those things. So let's get rid of all that stuff and start from scratch. Because if you're not breaking down and absorbing your food, what makes you think you're going to do a great job absorbing all of this,
Exactly.
all the supplementation you're putting in the system? And why would you think it's going to work the way you think it is? It's a nice idea and a nice strategy to think that we can control it. But the analogy I typically use for my patients is when they say, well, I thought if I took it, it would do this. And I always say, do you have kids? And they say, yes. I said, have you ever told your kid to go clean their room and you came up an hour or two later and wasn't done? And they're like, sure. I'm like, well, why didn't they do it? I don't know. I'll tell you why. They didn't want to, right? They get to make a decision when they go to the room, whether they want to clean it or play video games or watch TV, right? When it comes to the vitamins and the nutrients we force into the system, it's naive of us to think it's always going to work the way we want it to. Like, hey, I want you to take this. It's going to support your methylation. It might support your methylation, but it may not. It may do something totally differently. And that's sometimes when it doesn't do what we want it to do, that's when people say, well, I can't take B6. I'm allergic to B6. I can't take B12. I'm allergic to B12. No, if you took something, if you were allergic to B6, you wouldn't be able to eat food because it's in your food. You're not reacting to the food. You're taking copious amounts, probably super physiologic doses of supplements to try and do something that the body does not want it to do. That's my take.
Exactly.
So let's talk about
one thing that you mentioned,
sure.
Which is correct. Just because they have an MTHFR mutation, it does not mean that they need folate. And on the same token, if they don't have a mutation, it doesn't mean the methylation is working perfectly. They may still have a problem with methylation if the body is not working well. So we need to investigate and find out, what's going on with the body and what they need.
And that's exactly what my next question was going to lean towards, which is it just because you have I want to talk to you about how we assess if that genetic polymorphism is an issue or not. But that's a really important point to the people listening is just because you have a polymorphism does not mean that is your problem, right? Everybody's got polymorphisms, tons of polymorphisms within their genetic load. Doesn't mean that every polymorphism is a problem. Many times there's a polymorphism, a change in the gene here, and then 3 or 4 genes down the line from that gene, there may be another polymorphism that may compensate for the first one. So just because you have a polymorphism doesn't mean it's the cause of your problem or you necessarily need to treat it. We don't treat the gene or the genetic polymorphism. Our focus is on the patient, not the gene. That's the way it should be.
Now, because methylation affects almost every aspect in our body, what I say is that most of the time, methylation might be part of the problem, but most of the time is not the only problem. There's a variety of issues that may involve methylation, and in some case, methylation is the main issue. In some case, it's just a minor issue, and you have to look at what. Usually, most of the time, there are several things affecting the person's health, and we have to look at everything. The only thing is methylation is also one important factor that shouldn't be left out. So you should look at everything else, including methylation, and find out if it's a factor, if it's not a factor, and if it is a factor, how much of a factor that is. Maybe it's a small factor, maybe it's a big factor, but we just have to look into that and not forget about it. That's an important factor to
Exactly. So let's talk about what methylation is, because people may be going, I don't even know what that is. So what's a methyl donor and what's methylation?
I'm going to leave all the biochemistry terms and explanation for you, but I'm going to give the general. Methylation is a complex biochemical process that controls gene expression, turns genes on and off, controls intracellular detoxification, has an effect on the immune system, on DNA, RNA formation, and several different other functions. Go ahead and give the specific technical explanation. English being my second language, sometimes I forget technical terms. It's hard, but you speak beautifully, and you're helping out to explain what I want to say, because you understand about the subject.
So when we talk about methylation is the process of transferring a carbon, which is back to your basic science class, a carbon and 3 hydrogen molecules are attached to it. We call that a methyl group. And when we transfer that methyl group from one substance to another, that often creates chemical reactions. And it's really important, as you said.
Methylation attaches a methyl group to homocysteine and converts homocysteine into methionine. So Dr. Lynch, since you mentioned, he says methionine could be called methyl homocysteine, because it's just is the attachment of the methyl group that converts from one form to another.
And so we need that. We need folate, we need B12, we need a bunch of other things to allow for that methyl group to be generated and then to be transferred. And so that's what methylation is. And we use it for a lot of different things: detoxification, neurotransmitter production. There's so many things that it's involved in, lots and lots of things really important. And so when we're looking at somebody's overall physiology, and we're considering maybe they have an MTHFR or any other gene polymorphism that's involved in some of these cycles, we want to look at that and say, okay, we have a gene polymorphism. How is it potentially impacting this person's physiology? So if somebody wants to know if they potentially have a methylation problem, that maybe be impacting their neurotransmitter physiology, maybe impacting their energy production, maybe impacting their detoxification processes, what should they be looking for from a lab perspective and testing perspective? Now, the individual person might not be like, well, I don't know what to order, but we should mention some of the tests or lab testing that might help clue somebody in that maybe they're having some problems with their methylation processes. So we could talk about what labs, and we could talk about maybe common signs and symptoms.
Absolutely, yeah. Well, common signs and symptoms, I get these questions all the time, and because methylation affects your whole body, so you can have a huge array of symptoms. So there isn't, sometimes people ask, what is the symptom of MTHFR? They say like that. What's an MTHFR symptom? Or what a methylation? Well, it's not like a methylation symptom or MTHFR symptom. However, I can say that it's very common that we can see fatigue and brain fog. These are two very common symptoms that we can find in people with methylation issues. I would say the 3 biggest ones that are most common: fatigue, brain fog, and anxiety. With that said, there are 20 other causes for these as well. If you have fatigue, there are several things that can cause fatigue. There are several factors, causes that can, cause brain fog or anxiety. So with that said, it goes back to we have to look at the individual and find out what what is really causing it. But these are common. As far as lab tests, one thing I want to point out, for example, a lot of traditional doctors, they just look at homocysteine because of the connection with MTHFR mutation raises homocysteine. So that's the only thing they do. If the homocysteine is high, you got a problem. If the homocysteine is within normal range, you're okay. But this is not the best marker for methylation. There's another gene, for example, we all know that, most practitioners know that MTHFR will raise homocysteine. But there's another gene, for example, the CBS gene, the cystathionine beta synthase gene, CBS C6990. Then if you have a mutation, it will lower homocysteine. So, for example, you may look at somebody and the homocysteine is normal, but they have a MTHFR, a double mutation on the MTHFR C677T that would cause homocysteine to go up, but they have the CBS double mutation on C6990 will cause homocysteine to go down. So the homocysteine will have a false normal. It will look normal, but you're still not methylating. The patient may still not methylating well because instead of converting homocysteine into methionine, the CBS gene converting homocysteine into cystathionine. So the methylation wheel is not turning the way it's supposed to. You understand? The homocysteine will look normal, but the patient is still not methylating. So I look at a homocysteine as a valid marker because if it's high, we know there's likely a problem. But I like to keep things simple. I look at folate. Most of the time, I'm looking at the serum folate. You have you're gonna other tests. You look at the folate inside a cell, which is better. But most of the time, I'm looking at serum folate, and then I estimate based on the MTHFR what they are able to metabolize. Look at homocysteine, and then we'll most of the time I'm correct with that. We give the right prescription with methylated folate. If they need optimize the gut, always got to work on the gut. And I'll say 90% of the time I achieve results with that. But I would like to hear all the labs that you like.
Well, if we're going to look at methylation, I think, homocysteine is one marker that you can look at, but as you said, it doesn't really tell the full circle. So if you're a person who's, for the listener, if you're, hey, I know what MTHFR is, I know what homocysteine is, I think I have a methylation problem, I've been told I have a methylation problem. I think you have to look at more, as you said, you have to really look at more than homocysteine. And one of the challenges when looking at labs is not just looking at whether they're high or low, but interpretation, and which is what Ricardo's trying to explain through when he's talking about this. You got to look at multiple markers along with the patient signs and symptoms and say, okay, that's normal, but is it appropriate? Given what my patient's showing, is that value normal and appropriate? And that's what he was talking about. Like you might have an MTHFR homozygous polymorphism that reduces the recycling of homocysteine, so it can build up in the bloodstream. And you would, if you just saw that and you saw a normal homocysteine, you would say, okay, that's not having an effect. But the other part of what he was saying was, but if I have a CBS mutation or something that's draining my homocysteine down what we call the transsulfuration pathway, it may look normal. And so somebody who's not as astute at this may say, oh, it's fine. But this is where interpretation becomes. You can't just look at these lab values and say, oh, that's high, that's low, that's therefore, if it's low, I treat it. If it's normal, I don't have to do anything. If it's high, I treat it. We need to interpret. So we have to, anytime we look at lab values, what an astute physician is going to do or should do is they should say, based on your signs and symptoms and the person sitting in front of me, is that lab value normal and appropriate? Is it normal but inappropriate given my patient's current state? Is it abnormal, totally appropriate given how my patient's presenting, or is it abnormal and inappropriate? And that's what should be done. And for a lot of people, if you're just going to a physician who's saying, hey, it all looks, it's in the reference range, they're not doing a great job. They're not interpreting labs, they're reading labs. And that's where
Which
people
most of the
fail.
time that's what happened. They
Absolutely.
they just scan to and they say, okay, everything looks good, everything's within normal range, you're fine.
And they may never even look at it,
Yeah,
right?
exactly.
The assistant may look at it and say all the labs were normal, but this is why I emphasized before, if you're going to get a genetic panel done, make sure, if you're getting it done, make sure you're working with somebody who understands what it means so you don't just start treating gene polymorphisms and having some potential challenges. And so there are other tests out there. There's tests like Doctor's Data that looks at a little bit more of a methylation panel, but probably my favorite is HDRI Labs comprehensive methylation panel, where they look at the full folate cycle, the full methylation cycle, the transsulfuration cycle. They look at reduced and oxidized glutathione. So it's a really comprehensive panel. So if you take that test plus your genetic panel for the folate cycle, the methylation cycle, the transsulfuration cycle, and your regular labs, now you've really got a lot of data to see where potential breakdowns occurring. So we can do it simply. We don't always run all those panels on somebody based on signs and symptoms and how they feel. We might already say, well, I already know your methylation is compromised. Your estrogen levels are high. This is high. We already know that they're compromised. We don't really need to do any additional testing. We can make that assumption based on what we see and what your signs and symptoms are. But there are a couple ways to do that. And there's a couple labs that do even more comprehensive tests to take a look at. They're going to cost a little bit more money, but I think if in general, I like the simple strategy. You interpret, you say, there's probably some problems here with these in these pathways. I'm going to work on the gut to get better absorption, make things better. But if you're doing a lot of the right things and it's not going the way you want, one of those more in-depth, comprehensive methylation panels can be really helpful. So
yes. And
I want to touch base on a couple things here in regard to these things. And one of those is when we talk about methylation, the strategies are typically, if somebody's got problems with methylation, you'll typically have people say, hey, you need to take methylated vitamins because you have a polymorphism of MTHFR, and you have to take methyl B12. And some people do well with methylated vitamins, some people do not. What do you see in you working with patients? Do you see people having challenges with methylated vitamins, and why do you think that might be?
It's interesting because even though in theory, people with certain mutations could have more problems with certain vitamins. For example, people with COMT mutation, they may not respond well with the methylated B12, the methylcobalamin they call, and then you have to give the hydroxycobalamin. But clinically, what I have seen, at least my experience, is 90% of the time they respond fine with methylated vitamins. They don't have an issue. So it might be my geographic location, or I'm not sure why, but my clinical experience, I really didn't see much of a problem with methylated vitamins with the different mutations. On occasion, I do see patients that they cannot tolerate a methylated vitamin at all. They have to take very small dosage because they just throw their body way off. But it's not common. And I've seen those and I remember Dr. Lynch mentioning about his experience with that they have to go very, small. And I've had some of those cases that you have to be very careful and give very small dosage. But most of the time, they handle fine. So I don't think it's a big issue. And if you do take methylated folate and you react to it, and then you stop and take a little bit of niacin, and it's an antidote for a methylated folate. You learned that from Dr. Lynch. But I would like to hear your experience with people reacting to methylated vitamins.
Well, there definitely are people, and I've seen people that have come to me that swear they have methylation. They can't take anything with a methyl donor because it has problems. Um, there are people. I think there are people that have challenges with it, and my perception of why somebody might have a challenge taking excessive methyl donors is one. Again, we're assuming that just because we give a methyl donor, it's going to do what we want it to do. So for the
Mhm.
listener, when we're talking about methylation reactions, typically we're talking about enzymatic reactions, making chemical reactions work. And just because we give a methyl donor doesn't mean it's necessarily going to do what we want to do. Matter of fact, we can, if the methylation cycle is backed up, if it's clogged, we may give a bunch of methyl donors and it may be potentially problematic. And methylation reactions may not occur the way we want them to. But I think the other thing that we don't take into consideration is that methyl groups don't just do these chemistry reactions. Methyl donors can actually inhibit genetic replication. We have a gene, we have DNA that has to unwind so we can get at the gene, so we can replicate that, and then we can turn that gene and genetic expression on and have that enzyme work. The methylation group can help with that response and that reaction, but methyl donors can also turn off the unwrapping of some of that genetic material. So I wonder sometimes when everybody's methyl donors, methyl, you need all this methyl donors, is it potentially inhibiting some of the genetic reactions? We have, for the listeners, we have acetylation reactions that kind of unwinds the DNA, and then methylation kind of keeps it from unwinding. So just because we give a methyl donor for a chemistry reaction to occur, is it always doing that? Or could one of the reasons that people don't feel well on excessive methyl donor supplementation is because it's actually inhibiting some genetic expression and action? I don't know for certain, but that would definitely be one of my thoughts. And then
I
go
wanted to
ahead.
bring up a couple things. Now that you mentioned that I don't want us to run out of time. I wanted to bring that up.
Sure.
One is uh, with how much to take. I remember hearing a talk from Dr. Laskin. He's a researcher. He was doing a research with UCLA and Johns Hopkins with cancer.
Mhm.
And what they were doing is they're trying to suppress methylation on cancer killing cells.
Mhm.
And his philosophy was because methylation controls gene expression, and there are cancer killing genes and cancer killing cells that when you are methylating, you're suppressing the genes, but you will also suppress the cancer killing uh, genes and cancer killing cells. So they were developing this drug to suppress the methylation that will suppress the expression of those cancer killing genes. And it comes to my, my observation on that and my studies suggest that the treatment for somebody that does not have cancer is different than the treatment of somebody that has cancer. We should be more careful on strength and methylation. If somebody has cancer, we need to to be on the safe side, keep methylation on a much lower functioning than if you overstimulate methylation. You don't need to go into over methylation. Maybe you're still methylating well, but if you're on the high end, you may suppress those cancer killing genes. So that's some food for thought that I would be careful. If somebody has, I would say you want to make sure methylation is working, but keep it on the low end of the spectrum as opposed to, because methylation fluctuates and you can easily go into over methylation. You can have peaks. So what do you think about that? That's one thing. Here I am asking you the question,
Yeah,
whether,
well, I
you
think
know,
that I think that's really important, and I'll tie that to thyroid physiology. Um, But I think the important thing is there in the American way is if some is good, more is better. And that's not necessarily the case with human physiology. We can't really force the human physiology to do what we want it to do. And for people who are listening, I think the best place, and you've probably heard me say this multiple times, instead of thinking of your physiology like it's broken all the time, something's not working on, my body's not working. Change your philosophy on that from my body isn't broken. My body's trying to protect me from something. The body is this amazing piece of equipment that developed from two cells, amazing amounts of intelligence inside the cells and tissues. And the body wants to thrive, it wants to survive. But if it's being threatened, it's going to adapt. It's going to adapt. It's going to change. At the cellular level, it's going to change the way it works to deal with the threat. So anytime that something's not working appropriately, i.e. methylation seems to be compromised, we should do what we kind of talked about before is why might this not be working? Why might the body be adapting this way? Is this just a genetic polymorphism, or is there something else causing the body to behave the way it's behaving, right? And I think that's a better angle to come at this from, because if you just say, oh, methylation's not working, I'll just give a whole bunch of methyl donors, or I'll just pile the body with B vitamin or B9 or B6, you may not feel well because the body may be not having a methylation as at full tilt for a beneficial reason, not always just broken physiology. And we can tie this to thyroid physiology and cancer, because it's been my premise for a long time. I talk about it in my book, The Thyroid Debacle, that I always looked at thyroid physiology. Initially, when I was learning about thyroid physiology and what caused hypothyroidism, and we learned kind of an immune system out of control. And then we started seeing that, hey, we're not converting T4 to T3 well. And people were saying, well, that's broken physiology, so let's just give T3. None of that really made sense to me. And then when I read Navio's paper back in 2015 or 2016, when Ben sent it to me, that was my aha moment that I said, okay, maybe what we see in thyroid physiology isn't all broken. Maybe the reduced conversion of T4 to T3 is an adaptive response to cell stress. Maybe the thyroiditis isn't an immune system out of control, but driven by what we call pattern recognition receptors in circulation binding to the thyroid cells. And that those ideas and thoughts were then proven out by looking at all the literature and research that's out there. But when you look at it from that perspective, I talked to my co-author, Kelly Halderman, about this before we wrote the book, or in the early process of writing the book. I said, if this is the case, if the premise is right, that what we often see in people who are hypothyroid is it's not broken physiology, it's adaptive. The body's adaptively downregulating metabolism to deal with the threat, the thing that's more important. Then what happens when we just force more thyroid hormone into the system? Could that create more oxidative stress, more abnormal tissue development, more problems? Because T4 can bind to what are called integrin receptors on the outside of cells and cause cells to replicate. And I said to Kelly, the interesting thing is, if that's true, then, and hypothyroidism is a protective response, then if we gave people who are in this adaptive response to T4 therapy when the body really didn't want it, wouldn't it increase the risk of cancer in people who take thyroid medication? And she's like, wow, yeah, that sounds like it could, right? And I said, but nobody would ever do that study. Nobody's ever putting that together. And yet in 2019, a paper came out that people who have been diagnosed with hypothyroidism and get put on thyroid medication have a higher incidence of a whole bunch of different types of cancers. And now in the last couple of years, there's more papers coming out saying, hey, wait a second, hypothyroidism may be a protective response against cancer. And so now everything starts to come full circle. So when you talk about, hey, if we just put more methyl donors into the system, is it always good? No, maybe not. Maybe it was the physiology wasn't broken in to begin with. Maybe that was an adaptive response. So we need to be careful, just like we need to do with thyroid physiology.
And one thing you mentioned, thyroid, I wanted to bring it up. As you know, most conventional doctors, they look at TSH and TSH is high, they give the T4 and all that. And and we functional practitioners, and then we look at the the whole array of thyroid markers, and maybe the issue is not, as you mentioned, is not the thyroid, maybe it's the conversion and all that. And I had a conversation with Datis Karazian,
Mm-hmm.
the Harvard researcher,
Mm-hmm.
thyroid expert, and I talked to him about it, and he confirmed that the thyroid hormone, the relationship between methylation and thyroid hormone activation, because the thyroid hormone is activating the nucleus of the cells, in the DNA of the cells, and it needs methylation, acetylation, and transferation.
Mm-hmm.
So if people have methylation issues, and a lot of practitioners, they're not thinking about it. So maybe everything is working well. They regulate conversion, regulate everything. But if they have a methylation issue, they may still not activate the thyroid hormone properly. And I confirmed that with Dr. Correza and said, can we see if the blood work, if this is happening? He says, no, you cannot see. So all thyroid markers are going to look good. And they say you will not be able to see. So if you're treating a patient and you optimize their thyroid, everything is fine, but they still have thyroid symptoms. And if you haven't looked at methylation, there might be the issue. Because if methylation is not working, they may have problem with the thyroid hormone activation. So everything may be okay, but they still don't activate properly. So they may still have thyroid symptoms and is not. So that's one area that it may happen, that I think we should look into that. And it may be a small percentage. I'm not sure, but it's definitely a valid area that we need to consider methylation when we talk about thyroid as well, to make sure that the thyroid hormone activation that pathway is intact. That is.
So yeah, when we're talking about the conversion of was he talking about the conversion of T4 to T3? That's a deiodinase reaction. Was he talking that he thought
No,
methylation
I'm
played a
talking
part there?
about it's already converted to T3. Now the T3 is going to be activated. The T3 is going to be activated in the DNA of the cell, and it needs methylation to activate the thyroid hormone, actually.
Okay, well, you're talking about when it's bound to receptors
Yes.
for that response to occur. He was saying that you need some methylation support to do that.
Exactly. And if you have methylation issues, it's not going to do that well. So you have enough T3, but you're not getting it.
Yeah, I'd have to take a look at that and see. I'd have to take a look at that and see that, but definitely there's a correlation between thyroid physiology and the MTHFR gene, and the functionality of that gene. T3 plays a big role in the conversion of folate, and you need the activated form of folate, or I'm sorry, riboflavin, to actually help with that methylation reaction. So if somebody does have
It's vice
reduced,
versa. The T3
yeah,
affects methylation,
so one
and
affects,
methylation affects the activation of
absolutely,
T3. Yes, so they're both
yeah.
important.
So one affects the other, and
Yeah,
that's again where it kind of goes back to, it could be either one of the other things could affect the other.
absolutely.
And sometimes if there is this cell stress, cell adaptive response, genetic replication and nuclear reactions are downregulated as part of the cell stress response. So if you're going to have cell stress, you're going to have decreased T3, you're going to have decreased genetic replication, and that may be part of that adaptive response, because we don't want to provide more building blocks for a threat inside the cell. So
And
this
then
is
it
where it
becomes a
all gets really,
snowball.
yeah. So this is where it gets all really complicated and crazy.
Yeah.
And that's why if you're going to, for the client, for the patient, if you're going to spend money on these tasks, right? This is why it's so important to have somebody who understands some of this chemistry, because it's complex, it's heavy, it's thick. You
Yes.
can't just read these things in a couple minutes and say, oh, I know exactly what to do here. This is it, it's about the interpretation. So if you're going to get a test done, it's going to cost you $99 to get a genetic panel
It's
and
not
a
enough. No.
report. You're
Yeah.
not going to get the benefit. You're going to need somebody who can really break this stuff down and say, yeah, this is an issue. I can see how it applies. And now we're going to look for the appropriate way to address it versus just, throw a bunch of stuff at you, pop and pray with a whole bunch of
Absolutely.
supplements.
A non-individualized approach is a big stressor to the body. And a
Yeah,
good analogy is it's like a mechanic. You take your car, they put a little machine there, and they says, oh, the computer says it's your spark plug that is bad. And then they replace it. And then a couple months later goes bad again. You go there, then we put the machine, oh, the spark plug again. But the good mechanic, you're not going to be treating the computer says, okay, why is the spark plug being burned? And
right,
then look and find out the cause and treat. So that's what you're saying is you look at the report and say, oh, you got to take this. No, no, no. You're just reading the machine. You need to find out why. You need to, dive deep and understand what exactly you need. It's not just by replacing the spark plug is the vitamins. You don't need that. You need to treat the,
why?
whatever is broken.
Yeah. And that's really what the root of functional medicine is. It's trying to do as much as you can to uncover the why. And sometimes it's pretty simple, and other times it's pretty complex. It's kind of, I always use the analogy of Donkey and Shrek going through the uh, walking through the field, and a donkey says, ogres are like onions. You have to peel back the layers, right? And so the same thing that we need to do with our clients is like, we got to keep trying to, okay, like you're just saying the spark plug's not working. Okay, but why is the spark plug not working? Well, because it's not getting fuel to the engine. Okay, well, why do I not getting fuel to the engine? Sure, I can continue to replace the spark plug, but I'll be doing that forever, right?
Exactly.
Yeah, methylation is not working. You're going to need a methylated vitamin.
Exactly.
Okay, but
Absolutely.
why is not working? Why is that
You need
going
to
on,
keep
right?
asking why
Yeah,
and get to the root of the problem, and then you treat everything accordingly, and then magic happens.
right. That's perfect. I think we'll leave it with the magic stuff. I think what I'll do is I'll get you, I typically do this. Um, what are, now that we had this complex conversation, somebody is maybe their head spinning now. What do I do with this conversation? If we had to summarize this up, three things that somebody who's got maybe some chronic health issues, what it would be the three tips of advice in relationship to folate, genetic testing, and methylation that you would recommend?
Okay, so couple things. One, I think the biggest thing is they need to get someone that really understands it to coach them, to guide them on the right direction, because it's a very complex issue. It's not for them to play with their own health trying to find out. I think that's the number one. And the other thing I want to say is that the three most important things for longevity. I place diet, which is very important, as number three. It's very important. Number two is exercise, which by research has proven that exercise supports methylation.
Mhm.
And the number one, be happy. Do something that makes you happy. Happiness it's really that important. So I'll leave you with that.
Yeah, I think it's good. I don't know that somebody said that was number one before, but I think that's an important one for the listener. Mindset has a huge effect on your overall physiology. When you're unhappy, when you're miserable, you manifest that as an upregulated sympathetic nervous system, a more fight or flight response that downregulates your parasympathetics. And now nothing is going to work effectively. I must have this conversation 5, 6 times a day with clients and say, hey, listen, the number one, the elephant in the room here is what goes on between the 6 inches of your ears. And
Mhm.
you've been through 5, 6, 7 clinicians and doing the same things over and over again. Maybe everything that's being treated is the effect, and the cause is really what's going on between your 6 inches of your ears. And that may be the biggest thing to work on, right? If you're unhappy, figure out why, right? Work with somebody to figure that out. But that's a huge part. I call that emotional or psychological fitness. It has to be really high if you want to get healthy and get well. So I think that's all great. Tell somebody like where they can find, what's the name of the website, and give them some idea of what they can find on the website as far as testing and maybe practitioner support.
Absolutely. The mthfrdoctors.com, and they can order the at-home cheek swab, the MTHFR test, COMT test, cortisol test. Lots of articles, good information there. Yeah, it's a good resource for patients.
And if they were looking for somebody that
There's a wellness coaching. They can sign up for wellness coaching and get help through like Zoom sessions and such.
And then do you have,
Oh,
is
they can.
there a list of practitioners as well?
I don't have a list of practitioners. They can have they certainly can buy supplements, although I suggest they get a guidance, but they buy supplements. There are many companies that I like and I endorse. I would like to endorse Dr. Lynch's company, Seeking Health. I have great respect and admiration for Dr. Lynch. He was my inspiration to get into MTHFR. And definitely you need to look for a company that has science behind it. And there are several, vitamins companies that I like. Seeking Health is definitely one of them. Good brand. So
awesome.
Yep.
Well, I appreciate you coming on the Thyroid Answers podcast. I'll let you know when this is coming out so you can share it with your friends and your family.
Absolutely.
And for the listeners, if you have any questions or comments or thoughts about anything we talked about, send any questions or comments to me, or put them below wherever you watch the video on YouTube. It'll be available, and this video will be, and the audio will be available on all podcast platforms. So once again, Ricardo, it was great talking to you. Have a fantastic day, and thanks for coming on the podcast.
Thank you so much.